Hi, everyone, and welcome to the next webinar in our Plantae Presents Global Plant Science Talk Series. My name is Katie Rogers, and I'm your host for today's webinar. Before we get started, I'd like to go over a few things just to make sure you get the most out of attending today's webinar. If you are experiencing technical issues, please let us know about those using the chat box or by emailing me at krogers@aspb.org. If you're having trouble connecting or need to leave the webinar early, know that a recording of this webinar will be made available along with all of the associated materials within a few days. If you have questions during today's webinar, please let us know about those using the GoToWebinar chat box. Today's talks will each be 20 minutes, followed by 10 minutes for questions. Jürgen Klein-Venn, who is an associate professor at the University of Natural Resources and Life Sciences in Vienna, is with us today to moderate your questions and read them aloud to our panelists. This webinar series is brought to you today by Plantae, the open online community for plant scientists powered by the American Society of Plant Biologists. I'd like to give a special thank you to all of our ASPB members who are attending today. Your ASPB membership dues help support and make these webinars possible. For any of you who have not yet joined ASPB, you can join today and use the discount code PRESENTS10 to receive a 10% discount on registration. ASPB members get early access to these seminars. You can learn more about ASPB and the opportunities that we provide at aspb.org. After this webinar, we will be taking a short break from this weekly virtual research talk series to shift gears and prepare for the Plant Biology 2020 Worldwide Summer. We are planning to start this series up again in September and welcome your feedback. If you haven't already done so, please take a moment to fill out our feedback survey. I'm going to leave the link in the chat box and suggest speakers. All right, with that, I'm going to turn it over to Jürgen to introduce our speakers and begin today's talks.
Thank you, Katie. I also would like to welcome everyone that is watching this right now, live or later on as a recording. It's great that we can finally host Caroline Dean and Jorge Casal in one session. And well, as Katie just mentioned, we are urgently looking for feedback to see how we can further improve this event. So during this COVID outbreak, we kind of kickstarted this initiative within 1-2 weeks. So it had to be fast, and certainly there is room for improvement. So let us know what you think, what we should do better. And first up today is Caroline Dean from the John Innes Center. Caroline did her PhD at the University of York. And after a postdoc in the US in Oakland, she moved in 1988 already to the John Innes Center in Norwich. Caroline is a Royal Society professor and was appointed Dame Commander of the Order of the British Empire in 2016. I really like this. So it's the second Dame that we have in this series together with Autumn. And Caroline received many more distinctions, and well, I will not go much into this. Just to mention the latest one, because she just got the Wolf Prize in Agriculture. And if you don't know this prize, it's often considered as kind of the Nobel Prize in agriculture. So if you're not yet impressed by her, by this notion, I'm sure you will be after her talk, because her highly elegant work is a real treat, and her research reveals how plants actually remember that they have experienced winter, and her group uses this readout as a model to understand epigenetic regulation in plants. Her talk is entitled Non-Coding Transcription and Epigenetic Switching of FLC. And Caroline, thank you once again for joining us today. And I invite you here to take this virtual stage.
Thank you, Jürgen. Well, hello, everybody. When I want you to look at this picture, I think it beautifully shows just how interconnected plants are with their environment. And this has intrigued me for many years indeed. Plants can't move around. They have to adapt to the surroundings, and to maximize their success, they have to change what they do at different times of the year. So the seasonal cues they use are photoperiod and temperature. And I've always been very fascinated by the temperature cues because they're so complex. The panel on the right shows a bird's eye view of what a plant feels. It's actually from a field in Sweden. But you can see the hourly temperature readings show that the daily fluctuations in temperature are enormous, almost as much as the whole fluctuation across the season. So how are plants reading those seasonal cues, temperature cues, and then adjusting what they do? And that question has driven my lab for many years now. And so we started off looking at the process of vernalization, the need for winter to align flowering with spring, so that we could explore these questions of how the plants were monitoring seasonal temperature. Now, we use Arabidopsis thaliana, and some Arabidopsis have this winter annual habit. They germinate in autumn, overwinter vegetatively, and then vernalization promotes the flowering in spring. But other Arabidopsis types, probably the ones that you mostly use, are these rapid cycling types. They don't need winter, they just cycle rapidly. So we started off many years ago now asking 3 different questions. Why do some Arabidopsis successions do this overwintering? If they do, how do they register these long periods of cold? And lastly, different winters, of course, are going to be different. And so how have different Arabidopsis successions adapted to those different kinds of winters? And we started off with natural variation, with 4 genetic screens, with these 3 different questions, expecting we were going to end up in rather different places to study. But no, in fact, all our avenues led to the study of this one locus, a locus called flowering locus C, FLC. It's a gene encoding a Madsbox transcriptional repressor that blocks the activation of genes that cause flowering. So plants that need to overwinter express FLC at a very high level as they germinate, and that determines this overwintering requirement. And all our analysis of that variation came down to how to quantitatively transcriptionally regulate FLC. So it came down to some very generic gene regulation questions. All the questions about how do plants actually sense that long period of winter, again through genetics, came down to actually studying a polycomb-mediated switching mechanism, and I'll talk about that today. And then the third question, in terms of how have plants Arabidopsis adapted to different winters, actually resulted in us studying natural variation of either the quantitative transcriptional regulation of FLC or this switching mechanism. So you can see all our roots came back to FLC. Now, if we look at FLC regulation through the life cycle in a winter annual, as the plants germinate, they express high levels of FLC and they remain vegetative. But as they are exposed to increasing weeks of cold, and I've just shown this as a block of blue for different weeks of cold, FLC is progressively shut off. See this northern blot at the top here. As the plants then move back into spring, FLC remains off. It's epigenetically silenced by this cold exposure. So all of the factors, the photoperiod, the ambient temperature, all of the other factors required to switch to flowering then function and the plants flower. But then as the seed are developing, what happens is that FLC is switched back on again. And as you can see from this FLC luciferase fusion included here, the contrast between this very slow shut off in the cold is that there's a fairly coordinated and rather rapid switch back on in the seed. Now, over the years of studying all of these different questions, it's taken us into these different aspects of this FLC regulation. All our studies of whether you need to overwinter or not as a plant is actually all about the quantitative transcriptional regulation, how high this expression is on this axis. For the rapid cyclers, of course, they start off already with a very low level. We've been looking at the need for cold, and that's taken us into looking at the cold-induced transcriptional shutdown, the epigenetic switch, which I'll describe, and then the epigenetic memory mechanism. And lastly, this epigenetic resetting in the seed. But today I'm going to focus on the cold aspects, the transcriptional shutdown and the epigenetic switch. Now, we've been working on these for many years, so what I'm going to do in the first couple of slides is give you a schematic of our current understanding of that transcriptional shutdown and epigenetic switching, I've got the references on the slide so you can look at those, but I want to get through that fairly quickly so we can get to more recent work on studying these, how plants are actually responding to the fluctuating temperatures. So the first step, the transcriptional shutdown by cold. Well, that took us into analysing antisense transcripts of FLC. They are shown schematically on the top right here. If this is the FLC locus, here's the exons in the boxes here. This is the sense transcript with the various exons. We found that actually in the cold, quite quickly in the cold, a set of antisense transcripts are expressed. They are, the promoter is at the poly-A site of the sense transcription unit. They are alternatively spliced and alternatively processed. And in fact, all our pathways keep taking us back to studying these different forms of these antisense transcripts. Now, when we first analysed and characterised these, and this is shown on this northern blot here, you can see how the cold increases their expression. The postdoc decided it would be great to have a name that everyone can remember. So these have been christened couleur. Couleur basically describes all the different forms of FLC antisense. Now, we've also found, actually by doing single molecule fish analysis, and this is what you're looking at in the panel in the middle, oligos either labeled with green for the antisense or red for the sense, looking at the nascent transcription of both strands, looking at the single molecules in nuclei, this is stained with DAPI here, we only ever saw either a green signal or a red signal. This is in this Rosa paper. So that showed us that there was mutually exclusive transcription at the locus, at each allele. And so therefore, if the plants are upregulating the level of antisense, that was a mechanism to help shut down sense transcription. So that's the first step in the, no, I can't move my slide on. Oh, there we go. That was the first step in the process. The second step that we defined was through the genetics, looking for factors required for this silencing of FLC during the cold. We did a forward screen and identified a need for polycomb repressive complex 2, which is a well-known chromatin modification complex, but also two what we now call PRC2 accessory proteins called VIN3 and VERN5. These are homologous proteins with these protein domain structures shown here. And these associate together in the cold and, guided by this DNA binding protein VAL1, nucleate at a specific site internally in the gene. Covers about 2 to 3 nucleosomes downstream of the transcription start site. So we have a polycomb repressive complex 2 with VIN3 and VERN5 at this site. And that's what happens in the cold. Now, it's a slow process because actually VIN3 induction is slow. You can see from the schematic, it takes many weeks to maximally express VIN3. But there are other reasons why this is slow too, and we're still looking into those. But the actual switch to the epigenetic state involves nucleation at this site, because if the plants are then taken back out into the warm, that allele which has the nucleation event remains stably off. Now, when the plants come back into the warm, in fact, this epigenetic silencing is reinforced by spreading of PRC2 across the whole body of the gene in the long-term epigenetic memory state. So those are the different stages we know about. And because we were analysing PRC2, which is classically involved with H3K27 trimethylation, of course, we looked at that particular histone modification over the locus in these different parts of the cold steps. Now, during the cold, this is what you see. You see an accumulation of K27 trimethylation at this nucleation site, which is just downstream of the promoter. Sorry. And you see that there's an accumulation of K27 trimethylation as the plants have increasing cold. This is actually during the cold. As you bring the plants out after cold, 7 days after, you can see that the K27 is now spread into the body. But in both cases, it's a quantitative accumulation of K27 trimethylation, which comes along with increased cold exposure. And early on, when we looked at this, we thought, well, this makes perfect sense. We have a quantitative reduction in expression matching a quantitative accumulation in our histone modification. But actually, and this is always true in molecular biology, you of course grind up populations of cells for your molecular analysis. This had given us completely the wrong impression. And in fact, through computational modeling and some validation experiments, what we now think of is that it's not a gradual increase, it's actually a switch. What happens is there's a transcriptional shutdown, as I showed you, and then the cells, and I'm showing you a schematic of the root here, are either in a state where they're epigenetically on at FLC, transcription's already off, they're epigenetically on, or they've switched and nucleated and they're epigenetically off. And I've shown a couple of red cells here to indicate these have switched. And with increasing cold, this low probability nucleation event occurs in more and more and more of the cells, in fact, at each allele independently. When the plants then come back out into the warm and the roots grow, I'm going to show you a real root in a minute, but I'm just explaining the schematic. What then happens is that cells that have switched, all of the daughters remain off. And the nice thing about roots is this anticlinal division means that this column of cells is a developmental lineage from this daughter down here. And so as you can see, as more and more cells nucleate and then they grow out after the transfer to the warm, more and more of the cells have switched FLC off in an epigenetically stable state. But if you grind these up and look at the FLC expression, it looks quantitative. But it's not quantitative at each locus, it's a digital on and off with the population average changing, the proportion of cells switching. And what you see in the bottom right here is actually what real roots look like, expressing an FLC luciferase with the gene still on. You see it in all the files, but with increasing weeks of cold, you see now progressive silencing of the files. So we had a switch. So if I just recap what we had found so far, is as the plants, first of all, as they're transcribing and making high levels, that also involves the antisense. I'm not going to get into that, but then they start to feel the cold as a transcriptional shutdown because of increased antisense transcription, which is a pulse. Then you have this nucleation independently. It's local at each allele, cis-based digital nucleation, giving a metastable memory for about 10 cell cycles. And then once the plants come into the warm, the PRC2 spreads across the locus such that that gene stays off now until you have the resetting in the seed. So transcriptional shutdown, quite rapid, epigenetic switch, much more slow, but that of course is important that it's slow because the plant has to perceive the whole of winter. Now, that was a summary of a lot of work schematically, but it means I can get back to actually now the interesting thing of like the block that I showed you of cold, we just thought of as cold, this uniform cold. But of course, as I showed you at the beginning, the plants are not perceiving uniform cold. It's a very fluctuating, noisy environment. How do they register these noisy signals? How do plants distinguish these short-term and long-term differences? And of course, for seasonal response, that's really important. Somehow they have to understand the memory of the previous exposure. So this took us into a whole set of experiments that actually the ERC happily funded. They like to have blue sky proposals, and we certainly, this was a very blue sky one, sort of a molecular biologist proposing to do field experiments. We were very fortunate to have Svante Holm, who helped us, and Torbjörn Saul from Lund at Swedish universities. And we then sowed a whole series of genotypes out either in North Sweden, South Sweden, or in a cannibalized greenhouse in Norwich. You'll see a mass activity of all our sowing, so that we could then put these lines out and harvest them every week through the winter. And in fact, we did some chromatin IP analysis too in Norwich. So what did we find? Well, we could actually monitor the temperature, and you'll see these are the traces in the different places in Norwich, in northern Sweden or in southern Sweden. You see winter looks rather different in different places. I'm showing you the data here for the FLC decrease, and that's plotted on a log plot, so you can see this exponential decrease because it makes it easier to look at. The data is this sort of fuzzy line, and the exponential log plot is the straight line here. And then this is VIN3 accumulation at the bottom. So on first glance, well, gosh, phew, at least it looked a bit like what was happening in our lab cabinets. I guess that was a relief that actually all of our analysis over many years in cabinets and greenhouses sort of made sense and happened in the field. FLC went down with increasing cold, Vin3 went up. But actually, when you looked at the slight differences were very informative. The rates of silencing were slightly different in different places. And actually in Norwich, it was very informative. So you can see here, this plot, this exponential plot here doesn't really fit the data very well. So if we zoom in on that one, you can see that Vin3 didn't accumulate for very many days from sowing. It happened to be a very warm autumn that year. And in fact, when we looked at the plot, it seemed to fit 2 rates better with a switch at the time that Vin3 was then induced. So this got us really thinking about what was driving accumulation of Vin3 and how that was influencing FLC. So I should say, and the 2 rates were elaborated further when we looked at what was happening in a mutant which didn't express Vin3, this accessory protein that went up in the cold, because actually it stayed at this slow rate, and it was only when you had Vin3 expressed that you switched to this higher rate of silencing. So we set up a series of experiments trying to match some of the conditions that we found in the field in cabinets that then could do step changes of temperature. So by analysing the different places and looking at roughly the different temperature averages, we set up 2 experiments, one where we had a 14-degree constant temperature each day. You had 14 degrees, or a step change in temperature up and down, going from 8 to 22 with an average of 14. So we call that 14 fluctuating or 14 constant. And similarly, we had a step around 8 fluctuating and also 8 constant. So this to try and understand which feature of the temperature the plants were monitoring to change to regulate their Vin3 and FLC regulation. And looking at the bottom right here, what you can see is that the 8 fluctuating, 8 constants is really very similar. But for Vin3, the fluctuating temperatures around 14 were really very poor at actually allowing Vin3 to accumulate. In contrast, and exactly the same logic, when we look at FLC, here the constant FLC, the constant temperature of around 14, FLC repression was much slower than a fluctuating around 14, showing that actually it was the low temperatures, it relied on the presence of the cold. So Vin3 needed absence of the warmth, and FLC repression needed presence of the cold. And the other interesting thing was when we looked at the expression of this antisense actually in the field conditions. This is the temperature plot. These are all of the genotypes and the FLC expression. There was one night where there was this massive spike of couleur expression. And when we mined in, and this little box here is blown up, it turned out that was the first night that the temperatures dipped below 0. There was a very strong upregulation of couleur upon that peak. So the picture that then emerged was that we had the plants perceiving different aspects of the temperature, and it was the fluctuations that they were monitoring. The low fluctuations somehow involved the couleur dynamics affecting FLC transcription. So to shut off FLC, you needed the low temperature. For Vin3 expression, for this epigenetic silencing for these modifiers that affect the PRC2 nucleation, you needed temperatures below 15 over long periods of time. But any temperatures above 15 suppressed Vin3, in fact, turned over all the Vin3 that was accumulated. And so this was a great way for a plant to monitor, have I got through autumn? Is it really winter? Because afternoon temperatures were often, looking at this, a feature of still autumn conditions. So it's a combination of all of these different phases that is driving the distinct aspects of vernalization. Now we're looking at the molecular features of some of these. We're looking at what the temperature effects on couleur. We've also been looking at this very long-term induction of Vin3, and I just show one slide of that. The paper's about to come out. This was again through a genetic screen looking for regulators of Vin3 that would cause Vin3 to accumulate slowly at these temperatures of below about 12 degrees. And we came across a gene NTL8, a protein This is a NAC transmembrane domain protein of a family that have been associated with many different stress responses in plants. In fact, the cold doesn't affect the transcript levels of NTLA8. It doesn't actually affect the protein expression. But when we look at a GFP fusion, what we find is in the warm, this protein is mainly expressed in the meristem. But in the cold, it accumulates. And through a great combination of actually computational modeling and experimentation, what we found is that this slow increase in NTLA8 accumulation is not an effect on its expression, but because it's a very stable protein, it's through reduced dilution due to slow cell division at low temperatures. And we would never have actually really figured all of this out without a combination of modeling and experiments. So in fact, Vin3 regulation is quite complex. There's a current temperature perception, there's this absence of warm, and then there's this long-term cold. That all funnels into regulate Vin3, which of course also regulates FLC, but then there's a whole series of Vin3 independent temperature inputs. So rather than major thermosensors, which is sort of what the field has been looking for over the last 20 years or so, we now view that there's thermosensing in a different way, that it is distributed throughout the regulatory network, and that each step is actually monitoring different aspects of the temperature profile. And it's the fluctuations that are driving a lot of that temperature sensing. And so we've been doing modeling to actually try and come up with a model that would predict exactly what's going on in the field. And I refer you to this paper. There is a model that can actually predict dynamics in field conditions. So let me just wrap up. You know, we started off looking at through the genetics, and that's taken us into these different phases of FLC. What I talked about today was the cold phase. So instead of thinking of it as this cold period, which was for a long time, you know, 8 weeks in the cold room, we now have to think of it in this much more complex temperature series where the temperatures are fluctuating, and it's the fluctuations that are driving a lot of what's going on. We've got cold-induced antisense transcripts involved in the transcriptional shutdown, but also we've got these fluctuations influencing accumulation of the accessory protein that's driving the nucleation event, which gives the epigenetic switch. And so we're continuing, there's so many things that come out of this work, but the two major areas of activity in the lab at the moment include the antisense mediated transcriptional plasticity, because that's affecting the quantitative levels in the warm as well as in the cold, and what's the basis of this actual switching mechanism. So we started from, you know, a very, very specific plant question all about transition to flowering and vernalization, and we've ended up in fairly hardcore transcriptional regulation, non-coding RNA, and epigenetic regulation. And I think a lot of the lessons we learn from FLC will be generic to a lot of perhaps environmental responses in plants, but also more widely in other organisms. Non-coding transcription is clearly now a big part of many fields. For, in our case, they're very important to shut down FLC transcription in the cold, but we see that that antisense circuitry is tightly connected and really allowing transcriptional plasticity of FLC in response to different environmental conditions. Once we have the shutdown of the transcription, very low probability it's then locked into this epigenetically stable state through this polycomb mechanism in a digital switching mechanism. It needs to be low probability because actually the plants need to make sure they've gone through the whole of winter. And it's all the cells contributing independently, making that decision of, you know, have I had enough cold exposure to know that winter has passed? And the other really significant change in thinking is that really temperature influences every step we've looked at. And it's the fluctuations really that are as important as the average temperatures. So let me just acknowledge these are the current people in the lab, courtesy of Zoom. This is our group meeting a couple of weeks ago. Of course, what I've talked about is work from many years. I've acknowledged a lot of those in the publications. Let me just point out Yusheng Zhao and Rhea Kurinotu, who have done the work on the NTLA8, which is just about to come out. And These are a fantastic group. It's actually two groups here because a lot of our work is done in collaboration with Martin Howard, who's a computational group. So everybody in red is part of Martin's group and everyone in black is part of my group. But we sort of work as a jumbo group and really embrace the computational modeling. These are our current collaborators and these are our funding. And I thank you for your attention.
Thank you, Caroline.
Thank you.
I will select a couple of questions here from the audience. Aman Husbands is asking about the cell type that actually can sense this cold-triggered epigenetic switch, and maybe extending on this one. So, which cell type is actually then required for the flowering effect?
Well, as far as we can tell from our fish, all of the cells shut off FLC with the antisense. And in fact, probably most of the cells actually nucleate. But in the schematic, I showed the meristematic cells because actually what's really important is which cells then have the memory of nucleation. And of course, as the plant grows out, it's only the meristematic cells that are going to be forming the rest of the plant, which has then got either the on cells or the off cells. So I don't think there's a specific set of cells that are nucleating, but it's important that the nucleation happens in the cells that then subsequently divide to form the rest of the plant in the spring.
Tess Davis is wondering, what is the current molecular views of devernalization in response to worms?
Yes, there have been a lot of papers in the 50s about devernalization. We haven't looked at that because when we looked in our initial mutants, and we were doing this in an FCA mutant, for example, or a Frigida plus line, it is quite hard to devernalize those. The publications that were, in the 50s were taking these very, very late natural accessions. And then I think maybe it's slightly different, but because we had done all our initial mutants in effectively early flowering backgrounds that we'd made a winter annual by adding this Frigida gene, it's hard to see. So we've not looked at that at all.
And Goodwin James is wondering about the correlation between Win3 accumulation and the decrease of histone methylation levels and how strict this is.
Well, we now have mutants where Win3 is ectopically activated and there's absolutely no correlation with the switching. So Win3 expression is clearly slow and nucleation is slow, but even if you give Win3, you don't nucleate and you don't then accumulate your K27. So there's many other steps apart from just Win3 expression that affects that modification.
All right. Wait. There are a lot of questions popping in right now, so I'm slightly losing the overview here. So Mike Stam has a question. So I have a question concerning the change in Win3 during the cold. Does the Win3 expression level fluctuates when the temperature fluctuates and gets above 15 degrees Celsius?
And
he's sorry, and he's also wondering about the. Yeah. Just go ahead.
We've looked at Win3 by fish, and actually the sort of quantitative changes, it's actually not a digital switching mechanism at Win3 at all. Its individual cells are graded in their response. We haven't looked so much at Win3 in the fluctuating temperatures. All we do know is that above 15, anything that has accumulated is lost. There's a lot more. There was a paper, a Hepworth et al. Paper that I referred to in the Nature Communications. There's a lot of detail in there on that work that I would refer the person to.
So Jonathan Villas-Boas was really fascinated by your field experiments and has rather an open question about climate change and how you see this as how it will impact on the flowering.
Yeah, well, this is one of the things that came out of the modeling when we tried to can we, you know, if you go back a bit, people have done vernalization models for crops for many years, where they've done a sort of day degree, they've mapped how many hours at certain temperature intervals, and then they predict what the flowering is going to be. In fact, they don't work very well when the temperature fluctuates widely. So if you remember, the 8 fluctuating and the 8 constant actually were rather similar to each other. The 14 fluctuating and 14 constant were very different. So in fact, the crop models work when there's no fluctuations. And of course, what's happening with climate change is much more fluctuations. But the model we've got now does actually manage to predict what's going to happen in these larger fluctuations. So that's a plug for that model. And actually, it's not all doom and gloom. If you read that paper, there's a bit at the end about, in fact, you know, there's a lot of buffering goes on. So there will be changes, but, you know, sometimes it's good and sometimes it's bad. It slightly depends on which climate you're looking at. But I refer you to that cell systems paper because there's a discussion of that in there.
Mm-hmm. Maybe also related to this idea, a question of Shipra Roel, and she wonders about the role of FLC in other plants, for example, in perennial flowering plants.
Well, George Kublin's lab have been looking in detail at this. They have been using Arabis alpina as a nice perennial system in the Brassicaceae, and certainly FLC plays a role in Arabis. So I would refer her to all of George's excellent work on that. We've done a lot of work in the annuals all the way through the Brassicaceae crops, and actually it's everything I've said is all true in all of those, except there are just many more copies in Brassica, and so each copy does a slightly different thing, and so it's a cumulative effect of different copies that give you the Brassica vernalization.
I was quite intrigued by your notion that actually the slow division rate upregulates the NTL8, and this then activates the Win3. So does it mean that actually if several factors interfere with the growth rate, that they would have a common impact here?
Yes, and we show that in the paper actually. The prediction is you change the growth rate and you would have an impact, and yes, they do.
Very nice. All right, Karin, thanks a lot for this very nice talk and also for the very nice discussion. And well, there were a lot of questions, and we will hand them over to you, and it would be great if you can still reply to those. And well, for everyone out there that is unhappy that I didn't pick the right question, and you would still like to continue this discussion with Caroline, please also turn to Twitter, use the hashtag Plantae Presents in combination with her Twitter handle, which is @CarolineDeanLab. Thanks a lot, Caroline.
Pleasure.
Next up is Jorge Casal, and now we're getting from cold to, let's say, warmer temperatures, and that relates not only to the topic but also to the fact that Jorge is our first speaker from South America, actually. And you know, when people were suggesting speakers from South America, his name was really always on the list. So we are particularly happy that he followed our invitation and is speaking today. And this also kind of nicely reflects how well he and his work is respected by the community. Jorge got his PhD at Leicester University in '89, and afterwards he took a position at the CONICET in Argentina, and there climbed up the ladder within this system. He's nowadays a professor at the University of Buenos Aires, and he works at the Leloir Institute, which is Argentina's top-notch center dedicated to life sciences. His group uses genetic and cell biological approaches to understand how light and also temperature affect plant growth and development. And I really love his title that he will give today. The talk that he will give today is this Shade-avoidance signaling warms up. And we are also warming up for you, Jorge, and please, the stage is all yours.
Okay, thank you, Jürgen, Katie, and ASPP for organizing these talks and inviting me to be part of them. Okay, let's get started by offering you a definition of what shade avoidance responses are. Actually, they are a set of growth and developmental responses that reduce the intensity of current shade and or the probability of future shade. And we mean future because even light reflected on neighbors is enough to elicit some of these growth responses. For instance, in the case of growth responses, we have the classical vertical shade avoidant responses, where the stem grows faster and that projects the leaves towards the upper portion of the canopy where there is more light. We can also mention the horizontal shade avoidant responses, where asymmetric growth of either the leaves or the stem projects the foliage towards the side where there is more light. And in developmental responses, such as accelerated flowering, the cycle of the plant may be finished by the time when shade becomes stronger. The aim of our group is to understand shade avoidance responses in a complex environment. By understanding, we mean learning about the molecular mechanisms, but also about the functional implications. And we talk about a complex environment because plants have to integrate fluctuations in light and shade, and they also have to integrate the information coming from light shade with information coming from other variables of the environment, such as temperature. As you can see in these pictures of soybean crops, shading crops is unavoidable. And that is so because in order to make the most of the resources of the environment, we have to grow the plants at a high density. So there is going to be some degree of shade avoidance in any crop. Is that good? Is that bad? Well, it's a balance. There are negative effects such as the increased investment of resources in non-harvestable organs, such as the stem, that may also increase lodging, which is a real problem in agriculture. But we have to bear in mind that there are positive effects because the plants within the canopy with shade avoidance normally get a more efficient light interception. And these positive effects very often overweight the negative effects. Let me show you one example. This is a sunflower crop grown at a commercial density, and you can see that by the end of the cycle, all the stems are more or less at the upright position. Here we can see a crop at a much higher density, and by the end of the cycle, we can see that the stems are bent. And a more quantitative analysis shows that one of the plants goes to the left, next to the right, left, right, left, right, left, right, left, and so on. And that is so because when one plant bends into one direction, it creates the light cues for the next plant in the row to migrate to move into the opposite direction. So there is a sort of light-mediated self-organization of the plants within the crop. And if we prevent this self-organization from happening by physical means, we can actually reduce the yield of the sunflower crop. So shade avoidant responses are present in crops and they are actually important for yield. What are the molecular mechanisms behind these changes in growth? Well, we know that when the plants are under sunlight conditions, the activity of the photosensory receptors, such as phytochrome B and cryptochromes, is high, and that keeps repressed the pathways that promote growth. Therefore, we have plants of short stature. However, when we have shade or neighbor cues, the activity of the photoreceptors is reduced, and that releases the action of the pathways that promote growth, and we have shade avoidance responses. We can describe the pathway in further detail by saying, for instance, that PIFs, phytochrome interacting factors, are activated by shade by different molecular mechanisms. For instance, in the case of PIF4, we can see here in confocal microscopy that there is an increase in abundance, that is a rapid response where the plants are transferred to shade compared to wildlife conditions. These transcription factors bind to a large number of gene promoters, including YACA genes, which are involved in the synthesis of auxin. Therefore, there is extra auxin synthesized in the cotyledons. That auxin travels down to the hypocotyl where it promotes growth. In addition, we have this pathway here, this regulatory pathway, which involves COP1, which is a E3 ligase, which targets to degradation in the proteasome some negative regulators of PEEPs. We can see here that under wildlife conditions, the level of COP1 in the nucleus is quite low, but in response to shade, COP1 accumulates in the nucleus. As a result of that, we can see here the level of HFR1 is reduced along with the duration of the shade event. And in the COP1 mutant background, that reduction is much weaker. So by the work from other laboratories, we know that HFR1 is one of the direct targets of COP1, and that supports this regulatory mechanism here, which converges on the action of PEEPs. We can see that at different levels. For instance, at the level of auxin, we can see that there is a promotion by shade compared to white light in the level of auxin, but that promotion is actually lacking in the COP1 mutant background. And we can also see the convergence at the level of hypocotyl growth here, or by looking at a number of direct targets of PEEPs. You can look at any one of these boxes because the pattern is more or less the same. You can see that there is a promotion by shade of growth or expression. That promotion is lacking in the COP1 mutant background, but it is restored by combination of the COP1 mutation with the HFR1 mutation, giving support to the pathway as a whole. So this regulation here is given a sense of duration of shade to the whole pathway. That works very well for the first hours on the shade, but if we go to look at a plant 24 hours after beginning shade, we can see that the level of auxin is already back to the pre-simulation values. The rate of growth is even faster than earlier during shade avoidance, but it is not due to more auxin. It still requires auxin, but there is no more auxin than under white light. The thing is that this persistent shade actually activates several pathways. I'm going to describe you just one of them here, which involves the action again of PEEPs and OXIA proteins. You can see here for the case of P4 that in the cotyledons there is a large increase in the abundance in response to the first hours of shade. However, when we go there and look at the plant 24 hours later, the levels of P4 are already coming down, and this might be one of the reasons why the level of auxin is already back to the levels observed under white light. However, when we look at P4 in the hypocotyl, at the very least, the levels stay as high as when exposed to the first hours of shade. And in the case of vascular tissues, actually, it is even higher. And by overexpressing P4 exclusively in the vascular tissues, we have shown that there is a promotion of growth, giving physiological meaning to this observation. In the same vascular tissues, we have the expression of genes such as IIA19 and 29. And here we can see that the expression of, for instance, IIA19 is increased, particularly when we expose the plants to persistent shade. And that extra expression observed in response to persistent shade here is actually completely lacking in the P4 mutant background, giving support to this relationship here. Actually, these promoters are direct targets of P4. But now, how is it possible that growth, which depends on auxin, is promoted by these OXIA proteins, which are negative regulators of auxin signaling? Well, the thing is that the promotion is indirect, because these transcriptional regulators actually reduce the expression of IIA 17, which is a much stronger negative regulator of growth. And let me show you one example of that, one result to support that conclusion, coming from experiments in Nicotiana bentamiana, where we can see here the expression of the activity of the promoter IIA17. That activity is reduced when we induce IIA19, and actually we can go a step backwards and induce P4, and P4 is also effective to reduce the expression of IIA17, giving support to this pathway to keep growth at a high level when the plants are exposed to persistent shade. Okay, a number of laboratories have observed a significant convergence of the responses to shade and warm temperatures. And we now know that here we have, by the way, here we have the same pathway I show you to work during the first hours of shade avoidance. We now know that there are several points here which are under direct control of temperature. For instance, phytochrome B is a temperature sensor. Second, the PIF7 transcript is also able to sense temperature. And third, the evening complex, which is involved in the control of expression of PIF4 and PIF5, is also able to sense temperature. So these pathways work in a rather similar way for shade and warm temperature. And actually, the phenotype at the end is very similar if we compare the effect of warm temperature with that of shade. Let me go to 5B as a sensor with a little more detail. Phytochrome B is synthesized in the PR form, which absorbs maximally in red light. Upon excitation, it relaxes into PFR. PFR now absorbs maximally in far red light, and it relaxes into PR. And the same holds true for the second monomer of the dimer. So we can eventually get here to the PFR homodimer, which is considered the active conformer of phytochrome B, which inhibits the growth of the stem. So when we look up here at the top of the canopy, incoming radiation from the sun, it has a relatively high proportion of red. The ratio between red and infrared is about 1.1. But down here, due to the absorption of red light by photosynthetic pigments and the selective transmission and reflection of far red light, here we have a much lower red far red ratio. Therefore, under sunlight conditions, the photoequilibrium is driven towards the formation of PFR, and the plant is short. But in the presence of naval cues, the photoequilibrium is driven towards the formation of the inactive conformers, and the plant has a taller stature. But it is quite obvious here that in addition to changes in red-far ratio, which actually we can't see, there are very obvious changes in irradiance if we compare what happens up here in the canopy with what happens down here under the shade. So we can ask, are these changes having any effect on the status and activity of phytochrome B? Well, yes, they do. How do we know that? Well, for instance, if we grow the plants under a high red-far red ratio and high radiance, which produce a high proportion of PFR, we can see that 5B fused to TFP forms large nuclear bodies. And we can reduce the size of these nuclear bodies either by reducing the red-far red ratio or by reducing irradiance. So if lowering the red-far ratio lowers the proportion of PFR, we might expect low irradiance to do something very similar. How is this mechanistically possible? Well, of course, you know that these are light-mediated reactions, but they cannot account for this irradiance dependency. And the reason for that is that we know that you need very little light here to reach the photoequilibrium, the proportion of PFR. So there has to be something else. Well, that something else is thermal reversion. Thermal reversion is the spontaneous reversion of destabilization of PFR, which goes back to PR. Is this thermal reversion important? Is it significant in any way? Well, here we have the PFR level, which we have actually estimated based on a cellular model of phytochrome B, and we have recorded the light at the top of the canopy and at different positions within the canopy to feed this model. And here we have the dynamics of PFR starting at the middle of the morning and towards the end of the day. For each one of these cases, we can see two lines defining an area here. The upper line is for the photoequilibrium, that is to say the proportion of PFR established just by these photoconversion reactions. And down here we have the steady state, which is the level of PFR established if we also take into account thermal reversion. So when we look what happens at the top of the canopy where there is a lot of light, there isn't much of a difference between photoequilibrium and steady state. Well, maybe a little bit at the end of the day, but nothing else. However, when we get deeper into the canopy where there is less light, the impact of thermal reversion is more and more important. And that is so because when there is a lot of light, these arrows here are so fast that thermal reversion becomes diluted. But that is not the case when there is less light and these arrows here are not that important. A significant conclusion or possibility after this is that, if thermal reversion is actually affected by temperature within the physiological range, well, the whole system of phytochrome would be affected by temperature within the physiological range, and therefore phytochrome would not only perceive sunlight and neighbor cues, but also temperature. And we were actually able to see that PFR levels measured spectrophotometrically depend on temperature within the physiological range by using phytochrome B synthesized in vitro, and also by using etiolated seedlings where phytochrome B can be measured. But let me show you the results based on plants which have the same age and growth conditions used for shade avoidance. And the experiment is based on something I showed you before. I mentioned before that if we grow the plants in the high rainfall region and high irradiance, which produce high PFR, we have large nuclear bodies. If we predict that the high temperature to reduce PFR, we might expect to also see a reduction in the size of the nuclear bodies. So we did the experiment, and actually, as you can see here, between 20 and 30 centigrades, there is a reduction in the size of the nuclear bodies. However, here in this range, it doesn't work very well. We don't see a reduction. On the contrary, the thing is that this is a bit more complicated. The size of the nuclear bodies will depend not only on PFR, but also on the rates of assembly and disassembly. And actually, temperature may eventually affect all of these variables. So in order to quantify the impact of temperature on the status of phytochrome B as PFR using the size of the nuclear bodies as a proxy, we compared the size of these nuclear bodies in transgenic lines where phytochrome, wild-type phytochrome used to complement the 5B mutation. We compare that with transgenic lines where we use a mutant form of phytochrome B, which doesn't show that reversion. So we compared the size of the nuclear bodies, again, as a proxy of phytochrome B activity for this purpose, and we got within the range of physiological temperatures for Arabidopsis growth, we have a significant impact of temperature on the status of active phytochrome. Okay, downstream phytochrome, of course, we have enhanced activity of PIFs. Again, P4 as an example here, it goes up in abundance not only in response to shade, but also in response to warm temperature or to the combination between shade and warm temperature. And there is also an increase in the levels of auxin, and one of the consequences of that is shown here in the results of an RNA-seq experiment, where we actually use not only warm temperature, but also shade and the combination of both. And the cluster of genes where expression is increased by both treatments, warm temperature and shade, includes overrepresented GO terms such as cell growth, which relates to this, response to auxin, which relates to this, but we also find response to gibberellin, which actually relates to this here, because stellar proteins are negative regulators within the pathway of gibberellins. And I'm going to show you in the last slides two results connected to the action of COP1 as an E3 ligase directly acting on DELA proteins. To analyze that, we actually used a mutant form of one of the DELA proteins, RGA. This mutant form does not decay in response to gibberellin. You can add a lot of GA and nothing happens. However, when we grow these plants under either warm temperature or under shade, we can see a large reduction in the levels of this mutant form of RGA. And this response is completely lacking in the COP1 mutant background. From additional experiments, we know that there is direct physical interaction between the members of the COP1 SPAC complex and DELA proteins, and that COP1 can ubiquitinate DELA proteins in vitro. Finally, we can see here in real time that when we transfer the seedlings from light to shade or from 20 to 28 centigrades, we have a rapid accumulation of COP1 in the nucleus. And in parallel with that, we can see now the wild-type form of RGA. In the wild-type background, genetic background, we can see a rapid reduction of RGA in response to shade, and a rapid rapid decay in response to warm temperature. Particularly, the rapid component of the response is completely lacking in the COP1 mutant background. So, supporting the action of this regulatory loop both under shade and warm conditions. Okay, there is a significant convergence between shade avoidance and thermomorphogenesis. We think that this molecular convergence actually has to do with functional convergence. Because if the plants are, let's say, scared about shade, they are even more scared to the combination of shade and warm temperature. And that will be because shade reduces the amount of light for photosynthesis, and simultaneously warm temperatures increase the rate of mitochondrial respiration, which could actually impose a threat on the budget of carbon for the plant. And with that, I would like to thank you for, the conclusions. First, shade avoidance responses have both positive and negative consequences for the yield of the crop. I'll show you some data about that. There are different molecular mechanisms that operate in shade avoidance at different time scales, but they are all converged to act on hormones. I have shown you data on auxin, on GA, but there are also effects on brassinosteroids, and molecular conversions between the effect of shade avoidance and thermomorphogenesis, in our opinion, reflects functional convergence. Thank you very much for your attention, and I would like to share the group of people I work with every day. Thank you.
Thank you, Jorge. So once again, I will pick questions from the audience. You know, you started with this nice illustration of bending responses in the field. So this triggered the interest of Jochen Long. whether this can be actually optimized. So instead of planting the crops in a row, whether a dedicated management of distance, well, maybe they would feel then the shade, but they would kind of canalize the growth. So what do you think about this?
Well, yes, that idea is not really new. There are some experiments. We haven't been working on that, but there are quite a few people working on changing the design, the arrangement of the plants in the crop. It may have some advantages for under certain specific conditions, but that really is a possibility. Yeah.
So Taras Pasternak is wondering how you define cell growth, whether this is really just a matter of elongation rate, or whether really the volume of the cell increases.
We define that for in the case of the hypocotyl is mainly the length of the cell, not the whole volume. Because the diameter, for instance, there we measure the length of the hypocotyl. The diameter of the hypocotyl very often doesn't change very much, and in some cases it can actually be reduced by shade instead of being enlarged. Mm-hmm. So it's the growth in length.
Mm-hmm. So Nathan Hancock was kind of motivated by your illustrations, and is wondering now about the cry signaling and whether this one is also sensitive to temperature.
There's no evidence so far into that direction. It might be, but we don't know. I mean, there's no evidence that cry1 or cry2 can actually sense temperature.
So Francesca Cardinale is wondering whether there's any interaction with photorespiration and shade response.
Not that I know, actually.
Mm-hmm.
No.
And Thomas Vane, so as you, I guess this relates a bit to the early and late shade responses as well. So whether you compared early auxin responsive genes to late auxin responsive genes in your setups.
Yes, we have done some of those experiments. Of course, there are some differences, but for instance, the auxin response is very, very conservative. Auxin-induced genes dominate both transcriptomes. So during the early shade, that comes mainly from extra auxin, but later on, it comes from enhanced sensitivity to auxin.
Mm-hmm. And David Horvath is wondering about then the different growth rates in the different conditions. So would initial shade induce a different growth rate than later shade responses?
Yeah, there is a sort of commitment to shade avoidance. So there is a very rapid response during the minutes. I mean, and during the first hours, you can actually easily measure that. But later on, the response is even stronger.
Mm-hmm.
That's the trend.
Mm-hmm. So Victor Sanchez is wondering about the influence of these signals, so both shade and temperature, on root system behavior.
Well, shade actually has some effect. It depends on how you actually simulate shade. It's a bit more complicated thing. But and temperature tends to stimulate the growth of the primary root in Arabidopsis. The mechanisms are actually not the same. They are not defined to the same degree of certainty that there is information available now for the shoot.
Mm-hmm. Hermann Höfte is asking to what extent temperature sensing by phytochrome B is related to phase separation in the nucleus.
To what?
To phase separation in the nucleus.
We don't know about that. No.
All right. So I was also wondering. So you have, so in principle, it comes down to the to some degree to the PIV regulation, right? So what would be actually then the contribution of basically the direct impact of phytochrome B to the PIV and the indirect via COP1? So why is there this complex system, and what kind of what is the result of this system?
Well, I mean, the contribution of direct sensing of temperature by PIV7 and by the evening complex has only recently been demonstrated. So the experiments had not been done yet. So we need, for instance, to do some genetics with multiple mutants to see how much of the signaling current moves from Phi-B through PIVs, or it's direct sensing by the PIVs themselves, and in what condition each one is more important than the other.
All right, thanks a lot, Jorge, for this very nice talk. So I thank both of you once again for these inspiring talks, basically covering cool science and hot topics, I would say. And I thank everyone in the audience for joining us today. As mentioned, we will take a short creative break, and certainly would appreciate your feedback. And I hope you will enjoy your summer, and see you back hopefully in September. And with that, I hand over to Katie.
Yep, I'd also like to thank you all for joining us today. And I'd like to thank our speakers and our organizing team, Jürgen, Benjamin Schlesinger, and Mary Williams. Please do fill out the feedback form. We would really like to hear from you with your suggestions for the series and suggestions for speakers. And we look forward to seeing you again in September. Goodbye, everyone.
Bye.